is the nucleic acids. And the nucleic acid, of course, is part of our DNA and RNA, so it helps us make up our genetic material. And the thing about our genetic material, specifically our DNA, it helps us to make the proteins. So I don't know if we'll ever talk about it, but the DNA encodes information for us to make certain proteins. So DNA, also called deoxyribose nucleic acid, is double-stranded. So there's two strands, as you can see from this picture here, right? Deoxyribose. If you look at that name, first of all, I forgot to mention this, but whenever you see an "ose" at the end, this is usually a sugar. Okay, so when we're talking about ribose, it's a 5-carbon sugar. And deoxy, what do you think it means when it says deoxy? It doesn't have an oxygen. Very good. This is our genetic makeup. While RNA is ribose nucleic acid, and RNA has ribose, and it also contains, which is different from the DNA, uracil, which is a nitrogenous base. I'll kind of explain it a little bit. The thing about RNA, most of the time it's single-stranded. Okay, so here we have, if you look at this, what do you have here on this left side here? Is it RNA or DNA? RNA because, yeah, one strand. Here we have 2 strands. Okay, and looking at this, if you look at this, deoxyribose, notice how there's this hydrogen here, while our ribose has that oxygen over here. And that's why it's called deoxyribose, because it doesn't have that oxygen there. So those are some of the differences. We also have the nucleotides here, where we have cytosine, guanine, adenine, and thymine for DNA. We have the exact same thing in the RNA, except for that thymine. Instead of thymine, they have a nucleotide called uracil instead. Okay? So far, so good. Kinda. So a nucleotide is the monomers. The DNA and RNA are the polymers. And the nucleotide itself contains ribose, like we talked about earlier, which is pentose sugar here. So here, it's like a pentagon, right? Pentagon meaning that 5-sided. Very good. So here, 1, 2, 3, 4, 5. 5-carbon sugar here. So here is the ribose. And then next to it, we have a phosphate. And if we look at this phosphate here, notice what kind of charge is it? Negative charge, yeah. So DNA is negatively charged then, due to that phosphate group. And then finally we have the nitrogenous base that's next to it as well. Here we have the nitrogenous base. Can you tell if this is a deoxyribose or is it a ribose? This one here? Ribose because, besides saying it on the right, if I was to cover that up, how would you know? Right? There's an OH here, there's an oxygen here. Well, if we look over here, is there oxygen there? No, right? If there's no oxygen, it's deoxyribose. Okay. And looking at these nucleotides, since it has thymine over here, so is it a DNA or RNA? If it has thymine? No, it's DNA. RNA has uracil, remember? It doesn't have that thymine. Another thing is that these are purines. You don't have to know, but these are purines. These are pyrimidine. Adenine always matches up with thymine, while guanine always matches up with cytosine. Okay, so in RNA, what will match up with adenine? It will be uracil. Very good. Not thymine. Does that make sense? Yes? Because RNA doesn't have thymine. It has uracil instead. Okay, so please note that adenine always goes with thymine, guanine always goes with cytosine. So those are the nitrogenous base. We talked about thymine and uracil. This law of complementary base pairing, this is because I've been saying that adenine always goes with what? Thymine, and guanine always goes with cytosine. It gives us this complementary base pairing law, meaning that whenever there's an adenine, thymine will always go to it, match up with it. This will help us to replicate our DNA. So if there's some sort of damage to the DNA somewhere, we can always match it up again. Pretend there was a damage over here to this thymine over here, so this thymine is not there. Okay, so for the DNA to repair itself, it's really easy because it knows what to match it up with. If this strand is still intact, then it just needs to bring in another thymine in there and it'll be fixed. It also helps with DNA replication. How it does it is by splitting this DNA in half, so you have two single strands, and you just match it up, right? Just like doing a puzzle piece. So adenine always matches up with another thymine. Guanine always matches up with another cytosine. Does that make sense, kind of? A little bit? Anybody out there? There are other nucleotides that you need to know. ATP, adenosine triphosphate. This is the universal energy carrier. So we already talked about this earlier on, how ATP is how we use energy. So it's made up of adenine, ribose, and 3 phosphates, as you can see, we'll sort of see down there. But this was adenine over here. Here's ribose right there. Kind of see it. And over here you have 3 phosphates. Remember what was the charge of the phosphates again? They were positive or negatively charged? Negative. They were negatively charged. And so that gives it a lot of energy, because if there's so much negative together, then it wants to repel that last phosphate. And so that's where it gets most of its energy. So ATP is adenosine triphosphate. What do you think ADP will be? Yes, adenosine diphosphate. Very good. Here is an inorganic phosphate. This is after this last phosphate leaves by itself. That's that inorganic. Why is it called inorganic phosphate? What does inorganic mean? It doesn't have, not water, but it doesn't have inorganic. What does organic mean again? Carbon and hydrogen, right? So inorganic means it doesn't have that. It makes sense. If you cleave this off and have a phosphate by itself, it's not connected to any carbon or hydrogen. So that's why it's called inorganic phosphate. GTP is very similar to ATP. Instead of adenine, you have a guanine instead. So just take note of that. It's also another energy molecule. Cyclic AMP is kind of important. This is a secondary messenger for cell signaling. We'll talk more about that later. But remember how we talked about that receptor was a protein on the cell membrane? And that's how it receives signals from the outside. And then on the inside, we use the cyclic AMP. It's called cyclic because it is making a loop on itself. Unfortunately, you can't see it because this thing is blocking it. But you just trust me that it's there. And then there's these 2 coenzymes, NAD and also FAD. NAD plus, I don't want to even try to say this word, but NAD plus is made from vitamin B3. And it is a coenzyme, and the reason why I'm introducing you to it here is, well, one, it's a nucleotide, but another thing is because it's part of our cellular respiration that we will talk about later. In FAD, same thing. So these participate in our cellular respiration to make ATP in the end. So I want you to know that these participate in cellular respiration, that they're both coenzymes. Coenzymes, we'll later learn, it helps the enzymes do its function. And so NAD plus with 2 hydrogen will form this NADH and a hydrogen ion, while FAD will, with 2 hydrogen, will form an FADH2. And this will become more relevant when we actually talk about cellular respiration. But for now, just know that they're there, kind of thing. You don't have to know them in that detail for now, but later on we'll go into a little bit more detail. So a super brief overview of the cell. The reason why I want to do this overview of the cell because it helps build up on what we'll be learning pretty soon. First of all, the plasma membrane of an animal cell is just this outer portion of the cell itself. And does anybody remember which biomolecule makes up the plasma membrane? Say again? Which biomolecule? Is it a carbohydrate, a protein, or a lipid, or a nucleic acid? It's not a carbohydrate. Not a protein. It's a lipid, okay? Yeah, which lipid? It's a phospholipid. So the phospholipid bilayer makes up the plasma membrane, okay? So in plants, there's an additional cell wall around it, but I'm just talking about the animal right now. So phospholipid bilayer make up the plasma membrane. Phospholipid bilayer meaning that it's made up of a phospholipid. It has that polar head and also 2 fatty acid tails. And that polar head and fatty acid tails, there's 2 layers of it. So there's one layer that goes like this. Polar head, 2 fatty acid tails, right? Head, tail. Okay, so this is one layer here. The second layer, same thing, but upside down. So we were talking about, well, first of all, are these heads hydrophilic or hydrophobic? Say again? What kind of head did I say? It was polar, right? What does polar mean? Do you remember? Polar meaning that that has a partial positive and partial negative. Polar meaning that it has a partial positive, partial negative charge to it, meaning that it will be able to. It's basically hydrophilic, basically. So this is actually hydrophilic. And that makes sense too, because inside and outside of the cell has water, right? Over here, the fatty acid tails, what are fatty acids? They're... Which biomolecule are fatty acids in? Yeah, it's saturated, unsaturated fatty acids, but which biomolecule? What's the big... Lipids, yeah. So is it hydrophilic or hydrophobic? It's hydrophobic. And that also makes sense too, right? Because now, if you look at it, there's liquid out here, liquid inside here, inside the cell, but here, there's little contact with water, right? Because it's hiding in between. You gotta think of this as a 3-dimensional picture, so all this in here is going to be hydrophobic. Okay, we just talked about this. Okay, the cytoplasm is just this region, this area from the plasma membrane all the way to the nucleus. So this entire region is the cytoplasm. It contains most of the organelles within the cell. One of those organelles is the mitochondrion. A lot of people say mitochondria, which is fine because that's plural for mitochondrion. This is an organelle that has two membranes, as you can see. There's two membranes. There's an outer membrane and an inner membrane. And within these membranes, especially the cristae is the folds that you see in the inner membrane. These folds are there to increase the amount of surface area. Whenever you see folds, it usually means that it increases the amount of surface area. And that increase of the surface area allows us to make more and more ATP for cellular respiration to occur. We have a matrix. The matrix is inside the inner membrane. So that space, the fluid substance inside there is called a matrix. And of course, in between the membranes, we'll have something called intermembranous space, right? It means between the membranes, the space between the membranes. Okay. The reason why I'm having all these precursor things is so that when we talk about cellular respiration, it's a little bit more into context. So it'll be a little bit easier to understand when we get there. Okay. I think we have a question here real quick. Won't have you guys answer this. I'll just view it. I'll probably have you guys answer it next time we come back or something. Acknowledgements, pictures. So that's it for